Hi everybody. Today we are going to study for clinical imaging. Here are some key references from Wikipedia. Clinical imaging means we use human body as the object of imaging. In this lecture, we are going to study about optical coherence tomography and photoacoustic and ultrasound imaging. And then X-ray or X-ray computed tomography and PET, positron emission tomography, and SPECT, single photon emission CT. And finally, MRI, magnetic resonance imaging. Okay, let's start with optical imaging. Optical means we use light for imaging. It is like a photography we take using our own camera. And here is an example. In chemical biology, we are not interested in the fashion of clothes, but we are more focusing on biological part. This image is from Wikipedia as a representative human being. In this image, we can see the phenotype differences between XX and XY individual. But this is only the surface part. The optical source of this image is visible light, and visible light cannot penetrate our skin. If we want to see the inside of our body, what can we do? How about this? After removing skin, it is much easier to see inside. We can see the muscles and fats, etc. The preparation technique for this sample is called as a plastination, developed by Gunther von Hagens. I guess you have seen this exhibition, or at least some of those posters. If you further go in, we may see the connecting part of the body and eventually bones. By the way, all the images and all the samples are dead. Now let's think again. How can you see inside of human body? And if possible, live and non-invasively. When light hits human skin, short wavelengths of light reflects on the skin surface. Some portion of the light may enter the epidermis and dermis, and they may scatter or reflect. Optical coherence tomography is utilizing this reflected light. Depending on the different depths of the skin, the reflected light may have different travel lengths. Also, different physical composition of the tissue will affect the reflected light. For this technique, the one light source is divided into two, and one half is given to the sample and the reflected light is collected. The other half of the light is used as a reference and compared with the reflected light. The phase of two light could be different, and the interference pattern can be used for image construction. While the technique is called as a coherence, actually the combining process of the two light beam is more proper to be called as an interferometer. The depth level could be adjusted by the reference light path length change here. To increase the penetration depth of the light, usually near-IR light is used, and the image can be obtained up to about a half a millimeter from the skin surface. Now let's think about the term, tomography. Let's say here are several objects. If you give light from left side, some light passes through and some light are reflected, and generate this kind of a projection image. This projected image gives us only some partial information of the original objects. Tomography is imaging by section or sectioning. Each section can give us the information of an internal part of those objects. If we can do three-dimensional construction without the physical sectioning, it would be cool. If it can be done by multi-image analysis by computation, it is called as a computed tomography, abbreviated as a CT. This is an example of an OCT image of a skin, and we can see this is sweat gland through the skins. Skin had a limited penetration of the light. Then, among our body, which part is physically open or transparent? We use our eyes for collecting light. That means eye is optically transparent. So OPT also can be applied to eye imaging. The entry part of eye is called cornea, and then the screen side we call retina. Similar to the skin imaging, the input light can be divided into two. One half can be used as a reference, and the other half collect those information of retina and combine it together and generate interference images. And this is a constructed retina image. Light is coming from the upside here, and fovea is a color-sensitive area with lots of cone cells are localized. Different layers of retina structure can be analyzed by OCT technique. And interestingly, the nerve fibers are on this skin. That means light comes through and all the informations are collected to this nervous system and then transferred to the brains. Have you seen other deeper clinical image? Ultrasound has been quite popular imaging method for human body. Similar to the optical imaging, there is another wave for input and also for the reflected wave. The wave is not electromagnetic wave, but ultrasound wave. Ultrasound has a shorter wavelength than our audible sound wave. The relationship of ultrasound and sound wave is similar to UV and visible light. Using this sonar wave, internal organ or fetus imaging has been obtained for decades. One partial reason is the ultrasound wave is not so harmful to human body, unlike UV light. In ultrasound imaging, this kind of ultrasound generator and also detector is closely contacted on the skin. Usually gel is used to make the contact very tight and also to remove any air layer in between. If there is any object in the light path, it can generate a unique signal for the image reconstruction. And using this technique, a little low resolution of 2D image could be obtained. Recently, utilizing the multi-angle image acquisition, 3D Ultra CT becomes possible. So, not only two-dimensional silhouette, quite realistic 3D image also can be obtained. The image quality is getting better and better, and you can tell if the baby is more looks like a father or mother even before birth. It would be also fun to compare the face of the baby before and after the birth. If the image can be obtained in real time, 3D plus one more dimension of time, 4D ultrasound image is also possible. With this level of imaging quality and time resolution, it would be quite easy to detect any defect or problem in the fetus. In comparison to optical imaging, sound wave can penetrate deeper in tissue, but resolution in general a little poorer. By combining these two modalities, photoacoustic imaging has been developed. In this technique, the input is the optical radiation, the electromagnetic wave. The target molecule absorbs the light and generates heat-induced expansion, and then it generates a sound like a screaming. The sound signal is collected to construct the image of original target molecules. The light penetration problem is reduced to half. The specific position of the target molecules can improve the resolution of sound-based images. So, photoacoustic technique can be used for skin imaging for deeper than optical imaging. Using pulse laser, target molecule is excited and thermal expansion can be detected by ultrasound detect. The naturally occurring light-absorbing molecules are hemoglobins in blood. And depending on the oxygenation status, hemoglobin has a different light-absorbing windows. For example, around the 680 nanometer range, light cannot be absorbed by oxygenated hemoglobin, then only unoxygenated hemoglobin can be selectively imaged. In other wavelengths, both forms can be detected. Utilizing this selective energy absorption, it is possible to map which part of the brain is more oxygenated than the other part. High oxygen means more energy consumption is happening, so more metabolically active area than the other part. Now, let's look at this projection image. It looks like a visible light image. As a visible light cannot penetrate our body, Even though we take multi-angle images, at best we can generate only surface three-dimensional images. What if we use X-ray? X-ray penetrates soft part of our body, but cannot penetrate bone or metals, such as ring, so the projection images looks like this. We can see bones. If you take multi-angle images, through computational process, we can build three-dimensional images. This process we call computed tomography or CT. As X-ray CT is the most popular clinical CT imaging technique, if you hear just CT, usually it means X-ray CT. This is a constructed three-dimensional X-ray CT images. If you have a 3D image information by computation, you can also make sectional images by computation too. So this is a 3D model of the bone, and then other images are sectional images. In other words, tomography images. Now it is possible to see inside of a human body without physical cutting. So cool, isn't it?